Good morning, students. Good morning. Sorry, good afternoon. Students, can you tell me whether I'm audible and screen is visible? I will launch a poll. Kindly tell me whether I'm audible and screen is visible. Okay. Then we'll start the class. Okay, fine. Good afternoon, Neeli Shruti. Okay. Yeah, guys, in the today's class, we are going to talk about the plant growth and the development. Okay, you all know that plant physiology is a very important part in the botany, right? And from this chapter also, we'll get the around 2 to 3 questions. Okay, mainly the questions will come from the plant hormone here. So you have to focus more on the plant hormone. In most of the exam, the plant hormone question is asked. No, there may be a chances of getting the question from the other topics also. So that's why we are concentrating on all the topics today, but more on the hormone will concentrate. Okay, fine. So let us start that. Let us start a chapter. You all know that what is the plant growth and the development, right? When you want to plant any tree or you want to plant anything, means what you are going to do? You will take a seed, right? Yeah, you will take a seed, will select a seed, and that seed, are you going to keep, for example, that seed you are taken and you kept in any box? Is that going to be grow, guys? In the box, it is going to grow? Yes or no? Tell me. If you kept in a box or in a drawer, okay, or under the bed you kept, is that going to grow? No, right? Why it is not growing? Why that seed is not growing? You all know that, right? I think the topic you already know that you are doing the revision, but then also I want to take this part. It will not grow because that seed in the dormant condition, right? That requires some suitable condition to germinate, then only it is going to be grow, right? It requires soil and it requires water and it requires some temperature and the pH. Only in that condition it is going to germinate, right? Then it is going to grow, right? So these all the things you require. Not all the seeds is going to be suddenly it is going to be grow. The dormant seed, it will break the dormancy, then only it is going to germinate, right? Those all the things we are going to study today. And can you tell me, guys, the growth is mainly metabolic process, right? Either the anabolic process or the catabolic process. For the growth, we require energy or not? Or the energy is coming by the growth? We require, it is anabolic process or the catabolic process, growth. Remember, growth is a, growth is anabolic process, right? It is an anabolic process, means it requires ATP, right? It requires the ATP. Fine. Okay. Fine. Let us go to the seed germinations, guys. You all know that seed germination is a process by which a dormant seed, you all know the dormant seed, right? A dormant seed begins to grow and develop. Dormant seed begins to grow and develop into a new plant under the favorable condition. Okay, we require some favorable condition for the growth of the plant. Then only it is going to be grow, right? Favorable condition. And there is an activation of the metabolic pathway, the water absorption and emergence of the radicule and the plumule, the leading to the growth of the seedling. The mainly when the root, when the seed has been germinated, the first process is the radicule is going to be emerged. Then only we can tell that a germination of the seed has been started, right? You have to remember this, okay? And you know that a non-dormant seed is said to be at the quiescent stage. It is not going to be undergo the germination. It has to break the dormant seed, then only it is going to be undergo the germination. And you have to remember this point, the emergence of the radicule, not a plumule, okay? The emergence of the radicule is the first sign of the seed germination. If any seed is undergoing the germination, means the first emerging organ is the nothing but the, that is radicule. The radicule in the future, it will give rise to the root. Okay, fine. Okay. Let us talk about the types of the germination, guys. Here you can see there are the 3 types. One is the hypogel germination and apical germination. This topic I have taken because in some of the competitive exam, they have asked the question based on this, that they are given this diagram and they asked which type of the germination it is, or they have given the statement and they are asking which type of the germination it is. So that's why I'm giving this topic. Hypogel germination. In the hypogel germination, what will happen? Have you seen when you sow the seed, after growth of some time, the seed will come outside, right? The seed will, you can see outside, right? For example, you can see the bean outside. That type of the seed germination we will call as epigeial seed germination. The seed will come out of the soil. Okay, the seed will come out of the soil. That type of the germination we will call as epigeial germination. It is a type of seed germination in which cotyledon come out of the soil along with the encloser's plumule. That is called as that hypocotyl is going to be grow. That is called as epigeial germination. Epigeial, remember. Okay, the example is bean. Then coming to the hypogel germination, it is a type of the seed germination in which you cannot see the cotyledon, where the cotyledon was below the soil. Okay, that is inside the soil. That type is called as a hypogel germination, and the epicotyl will grow. And here the examples are the pea and maize and coconut. Pea, maize, and coconut. Only in the bean you can see the epigeial germination. In the some other examples also there, but we have taken the bean. Okay, fine. And vivipary. Vivipary. What do you mean by vivipary? Mean here the seed is going to be germinating when it is attached to the parental plant. When it is going to be germinate means when it is attached to the parental plant. That is going to be called as a vivipary. Okay, here you can see the example, that is the mango I have taken here. The mango plant, that mango is there in the mango plant, mango tree it is there. Okay, that mango has been formed, that mango is having the seed, right? That has been germinated. That has been germinated when that mango is attached to the tree, parental tree. Okay, that type of germination we are going to call as vivipary. Okay, remember that vivipary. Okay, the examples is the rhizophora and soneraceae. Okay, so this thing you can remember. Vivipary in mango we are given here. Okay, then coming to the growth, guys, you all know that growth is a irreversible, a permanent increase. Yeah, now we have grown, right? Now we want to join again to the 6th standard. Now we want to decrease our size and we have to again join to the 6th standard. Means is it possible? No, right? It is not possible because we have grown up, right? Now it is not possible to go back to 6th standard, right? So that is called as a irreversible and a permanent change, permanent increase in the size of organ and it's a parts of the individual cell. It is called as a growth. Remember always it is irreversible and it is permanent. Okay, and it involves a metabolic process that consumes the energy, means the anabolic process. Okay, just remember the statements. Then coming to the plant, because we are talking about the plant, you know that in the plant we will tell that a growth is indeterminate. What do you mean by indeterminate? Means here the plant will grow till its lifespan, till its life continues, the growth is continuing. But it is not in the case of animals. In the animals, the growth is determinate. After some time of the growth, we are not going to increase in our height, right? The weight is going to be increased. That is not related to the growth. Okay, the height is not going to be increases, right? That is called as the determinate growth in the animal. Okay, then coming to the indeterminate in the plant, that is mainly due to the meristematic cell, meristematic tissue that is present in the plants. Here you can see that there are two types of the meristematic cells are present. One is the primary and another one is the secondary. We will study about that. Okay, the way the growth is indeterminate in the plant, that growth we will call as the open form of the growth. Okay, that is mainly due to the meristems. Here we will talk about, in the plant we have seen that a primary growth, right? Mainly the primary growth is involved is increase in the height. Increase in the height of the plant. Where we can see the mainly increase in the height of the plant? In the root and in the shoot, right? In the root, there is a root apical meristem is there, and in the shoot apical meristem is there, right? That we are going to call as the root apical meristem and shoot apical meristem. They mainly helps in the increasing the height of the plant. Okay. Then coming to the another one, that is the secondary growth. Mainly we can see in the stem of the gymnosperm and in the dicotyledon we can see. Here, the girth is going to be increases, right? But we are talking about the primarily the indeterminate growth related is meristematic tissue that is in the plant. Remember this. Okay, then we'll go to the, and one more thing I have to tell. In the plant, there is a determinate growth is also going to be taken place. Where we can see the determinate growth, guys? In the plants, we are not telling the plant will show only the indeterminate growth. It will show the determinate growth also in some organ. Some organ of the plant will show the determinate growth. Can you tell me which are the organs, guys? I just want the answer from you where you can involve in the class. No, that's why. Can you tell me where in the plant a determinate growth is going to be happen in which organ? Just see, guys, imagine if the indeterminate growth is going to be happen in the leaves or in the fruit, what will happen? Are you going to see the mango of this size only or leaf of this size only? No, right? It is having a length of increased length of the leaves and increased length of the fruits. Yes or no? And flower also will be increased in any size. Right? Yes or no? The determinate growth we can see in the plants is in the flower and in the leaves and in the fruits. Those organs, those are showing the determinate growth where growth is limited. After that, it will not grow. Okay? Remember. Very good, Sahara. Yes, pa. Okay, that all called as a determinate growth in the plant. Then we'll go to the phases of the growth. Here, the phases of the growth is important. You have to remember the points, guys. Okay, there are three phases. One is a meristematic phase, elongation phase, and maturation phase. And here you can see that the root cap is helps in the perseveration of the gravitation. Geotrophism, we know, right? It is helps in the gravity. Where the water is there, right? There the root will grow, right? The root cap is helping in that. Whereas in the plants which are growing the water, right? Hydrophytes, there they are not having the any root cap. Okay, remember that. Fine. Let us their root pockets will be there in the plants where it is growing in the water. Okay. Here you can see three phases. After the root cap, you can see the meristematic zone. In the meristematic zone, the cells are there, right? That cells are having the rich protoplasma, more protoplasma, remember, and the large nuclei. Nuclei will be large. Please make a note of this. This is very important. And cell walls are primarily thin because they are undergoing the cell division continuously, right? So that's why the cell wall is very thin here. And cellulose. And there is a more plasma desmata connection is there between the cell. There is a more plasma desmata connection is there. Remember. Okay, what do you have to remember in the meristematic tissues? Those are having the rich protoplasma and they're having the dense nuclei and they're having the more plasmodesmata connection. Then coming to the elongation phase, after the meristematic phase, there is the elongation phase. The cells are now, they are very small, right? Just remember, now the baby has been born means later the baby is growing, right? It is nothing but the cells will come to the elongation phase means they are going to elongate. Okay. The occurs in the cell that is proximal to meristematic zone. The cell increased vacuolation. Remember, the vacuolation is increased here, vacuole. And the size of the new cell wall deposition is also going to be formed because it is maturing. Next step is maturing, right? So that's why a new cell wall deposition is going to be happen in the elongation zone. Then coming to the maturation phase. In the maturation phase, we will see the root hair. Remember, root hair we will see in the, here you can see this is the maturation phase, root hair you can see in the maturation phase, and this is after the elongation phase. The cell is attained the maximum size here and the wall is thickened, okay, and they have modified its protoplasma, okay. These all are the important points you have to remember. I think everyone know that meristematic phase, elongation phase, maturation phase, but you have to remember what is the cell inside, what is happening inside the cell. Okay, so here is the first question for you guys. Which of the following statement is not correct? Okay, not correct means you had to check the false statement. False statement. Okay, fine. The first one is the phase of the cell elongation of the plant cell is characterized by increased vacuolation. They are telling that the cell in the elongation phase, those are having the increased vacuolation. Now only we studied. Okay. The cell in the meristematic phase, the cell which are in the meristematic zone, those are having the abundant plasmodesmata connection, that telling. Then plant growth is generally determinate. Okay. Option D, the plant growth is measurable. Okay. So here is the poll I am running. Kindly answer me in the poll. Yeah. I'll answer the poll. Think once again, guys. Wherever in the statement wise question, which is not correct, which is correct. Sometime what we will do, no, we will just click on one thing, but after that we will think that, oh my god, this is not correct. Yeah. So that's why whenever you are clicking for the answer, whenever you are giving the correct answer, please think once again whether the statement are correct or not. I'm ending the poll. Yeah, all are not given the answer, guys. Kindly give me in the message box. Okay, thank you. So yeah, 50% given C and 50% given D. So the correct answer is option C, guys. Now only we studied right. The plant growth is generally indeterminate. Indeterminate, not determinate. Determinate in animals, right? Determinate in the animals. We will grow for certain period, then the growth is going to be stop in animals, not in plant. It is goes on increasing its shoot and root, right? The plant growth is indeterminate, not determinate, remember. And plant growth is measurable. We can measure, no, guys. We can measure the leaf, right? We can measure the plant, how much is the plant growth now. And after that 1 week, we can check how much growth the plant is increased. Right? Yes or no? Can we measure the growth of the plant? 50% have given right. Can you tell me, can we measure the growth of the plant, guys? Is it not possible to measure the growth of the plant? They are telling which one is not correct, false. Plant growth is measurable. Now I have planted the plant, right? Before 1 week I'm going, the plant growth is around like 4 centimeter. Okay. After I'm coming, the plant growth is 6 centimeter. I've checked the length of the plant. It is 6 centimeter. How much it is grown? Can you tell me how much it is grown? Before it is 4. Sorry. Yeah, before I'm going, it is 4. Now it is increased to 6. Okay. Now, how much it is increased length? 2, right? Now only you measured, right? 2 centimeter it is increased. Now you tell me whether the plant growth is measurable or not. Whether it is measurable or not. All are given wrong answer. Just think once again. Now, can you tell me whether the plant growth is measurable? Sahara, can you think once again? Here they are telling that which one is not correct. Here they are giving plant growth is generally determinate. Plant growth is generally indeterminate, not determinate. Have you understood this question, guys? Very good, Raju. Yes, Nili. Yes, Shreya. Just remember, plant growth is measurable. We can measure. Okay, this is also correct. And cells of the meristematic growth phase exhibit the abundant plasmodesmata connection. That is also correct. In the elongation phase, increased vacuolation. That is also correct. The wrong is determinate. The only one word is wrong here. Because in the plant, it is indeterminate. That's why. Sahara, did you understood now, Pa? Yes, Stephen, correct. Fine. Okay, let us go to the next question, guys. Okay, now next question, topic. Yeah, Sarah. If you have till doubt means kindly tell me. I will explain. Okay. Let us go to the growth rate, guys. It is an important topic. The growth rate, what do you mean by growth rate is what I told you now is that is nothing but the growth rate. Here we are comparing the growth with the time. Now only I told right before I'm going to the outside or the out for 1 week, the plant growth is 6 centimeter, for example, sorry, 4 centimeter, right? After I'm coming from the vacation, it is 6 centimeter. How much time I took is 1 week. Okay, 1 week I took. Okay, so here what I'm doing, I am comparing the plant growth Now the plant growth is increased to 2 centimeter, right? Before it is and after it is. What I'm doing, I'm comparing the growth of the plant with the time. Yes or no? With the time, I'm comparing. So the growth rate is nothing but comparing with the time. Okay. So what we are comparing? Growth. Okay. The growth rate is defined as increased growth per unit time. Unit time I am taken here 1 week. You can take 1 month or any time you can take. So the growth rate is defined as increased growth per unit time. Okay. An organism or its part can give rise to more cell or in number. Okay. When you take that growth in a unicellular organism, it is equal to reproduction. Right. Yeast. Yeast in 1 hour, it is divided to 2 cell or 3 cell, we can tell. Right. That is nothing but our reproduction also. But in a multicellular organism, it is division of the cell is nothing but the growth. Okay. Accumulation of the cell is happening, right? That is nothing but growth. Okay. Increasing the growth may be arithmetic or geometrical. Mainly in the growth, we can tell that there is the two things. One is arithmetic growth or the geometrical growth. Let us remember this. Here the equation is also important and the statement is also important. Remember. And the graph is also very, very important. Okay. In the geometric growth, what is happening guys? Here we can see one cell I have taken. Okay. This is the one cell. This is undergoing the mitosis. How many cell I will get, guys? How many cell I will get? 2 cell, right? I will get the 2 cell. So this is the one cell and this is the 2 cell. Now what will happen? This one cell is undergoing the maturation. It will undergo matured. Okay. This second cell is going to be divided again. The second cell undergoing division. Okay. The second cell is undergoing division. So this is the second cell undergone mitosis, right? Now what happened? This is a one cell and this is a 2 cell, right? And in the third, what will happen? We will get this cell I'm going to keep like this only. Okay. And this one cell is undergoing the maturation. I will keep like this. Okay. And now the second cell is undergoing the division. The second cell undergoing the division and 1 and 2. Okay. What is happening in the arithmetic growth is the one cell will undergo the mitosis and it will give the 2 cell. The one cell is going to be divided into 2 cell, right? The one cell is differentiating and another cell is undergoing the mitosis again. Okay. I'm able to hear, right? I'm audible, right, guys? I'm audible, right? Okay. Thank you. One minute, guys. I will tell you this again. Did you understood what do you mean by geometric growth, guys? Sorry. Did you understood arithmetic growth? See here in the arithmetic growth, what is happening? Here the one cell is there. This one cell is undergoing the mitosis means it will give rise to 2 cell. Right, this is the 1 and this is the 2. This cell is undergoing the differentiation means it is going to undergo the maturation. And this cell is going to be divided again. Now you can see I am taking this cell like that only. And this cell is undergoing the division. Now it has given the 2 cell, right? In this 2 cell, this is also undergoing the maturation and only this cell is undergoing the divide, right? Again, what will happen? I am taking like this cell I'm taking like this cell I'm taking like this, and this cell is going to divide, right? This is a 1 and this is a 2, right? This is matured, this is also matured, and this is also matured, and this cell is going to be divided again, right? And in this cell, one is going to be mature, and again, this cell is going to be divide 1 and 2, right? This type of the division, we are going to be this type of the growth, we are going to call it as arithmetic growth. One cell is keeping its meristematic activity and another cell is going to be divide. Okay. And here you can see we are expressing as LT is equal to L naught plus RT. What do you mean by LT? Yes, sorry, what do you mean by L naught? L naught means always tell that initial state, initial. Okay. L naught is the initial length during the initial uh, length before the zero time, and the length after the time, length at the time when you're checking, right? When you're checking at that time, it is 6 centimeter, right? Before you're checking, it is 4 centimeter, right? That is we called as LT. Okay. After checking, it is LT, right? 6 centimeter is nothing but the LT. Before it is 4 centimeter, right? That is L naught is nothing but the 4 centimeter. And R is the growth rate. Growth rate is nothing but 2 centimeter. How much it is grown, right? That is nothing but the 2 centimeter. Okay, fine. So I think you understood this. Let us go to the geometric growth. It is a quite common in the unicellular organism. You know that one cell is divided into two cells, where two cell is having the capacity to redivide, right? Meristematic activity is there. That is called as the geometric phase. Here you can see one cell is there. The one cell is divided means it is given to the two cells, right? Again, this two cell is going to be divided into two cell, right? Then again, this two cell is going to be divided. Each cell is keeping its meristematic activity. That is going to be called as geometric phase. Okay. So here you can see the graph. This is a time. On the x-axis, it is a time. On the y-axis, it is a population size. It will give the graph that is a sigmoidal. We will call as sigmoidal curve. Remember, the geometrical growth will give the sigmoidal curve. The phases are, first one is a lag phase. Whenever the growth is started, the growth will be lagging first. Okay. Then it is going to increase the size, increase the growth. So that's why always the first phase is lag. Then it will be log. And the third is going to be stationary. The growth will be not going to be increase or decrease. That is called as a stationary phase. Remember, one is the lag, log, and the stationary. And the equation for this is W1 is equal to W naught ERT. W naught ERT. Remember this. Okay. W1 is the final size, W naught as usual as the initial size, and R is the growth rate, and T is the time of the growth. And E we are taking, that is the base of the natural logarithm. Okay. And here you can see, guys. So this is the mother cell. The mother cell is divided into two cell, right? And again, it will give rise to the four cell. You can see like this. Two cell will give rise to the four cell. Then four cell will give rise to the eight. This 8 cell will give rise to the 16 cell. These type of the division we are going to call as geometric growth. Okay. Remember this. Okay, 1 minute. Okay, fine. I think we studied about the growth. There will be a growth rate, that is the geometric growth rate, and geometric growth and the arithmetic growth. And here you have to remember this is the point. An embryo initially shows an embryo first starting will show the geometric growth, because all the cell is going to be divided, right? The cell is growing now, embryo is growing, right? It's starting, it will show the geometric growth. Later in the life, it will show the arithmetic growth. Remember this point. Then we'll go to the growth, and now only in the question I told you, growth is measurable. Okay, growth is measurable. And in this, you have to remember, we can check the leaf growth. Now, before going for the vacation, the leaf length is this much, and after that, it will be increased this much. We can actually check the length of the leaf, and the weight of the plant also we can check. That is a dry weight, and the dry weight, and also the wet weight also we can check. And here, this is the important point you have to remember in this topic. The cell number in the maze. In the maze, you can see that a root apical meristem. This root apical meristem will produce a more than 17,500. Remember the number 17,500 new cells per hour, not per minute. Okay, per hour it is going to be increased. Okay, and you can see that watermelon, how much size the watermelon can increase its size, means it will increase up by 3,50,000 times. It can increase its volume. Okay, it is increase its volume. If they talk about volume, you can take a watermelon. If I talk about the number of the cells per hour, it is increased, means you can take a maize root. Okay, in the maize root, a meristematic cell we can increase per hour, that is 17,500 new cells per hour. Okay, this is important, you have to remember. Then coming to the growth, okay, there are two types. One is the absolute growth and another one is a relative growth. What you have to remember here is relative means comparing. Okay, here we are comparing the growth with the previous growth, that we are calling as a relative growth. Remember, absolute growth, absolutely how much growth has happened. Tell me absolutely means here you can see the total growth per unit time. Okay, here two leaves are there. If I am comparing the absolute growth for this means for the two leaves A and B means the absolute growth is 5 centimeters square. Okay, and here you can see the absolute growth rate for both the leaves is 5 square centimeter per given time. In the given time, for example, in 1 week, both the leaves grown for 5 centimeters square only. Okay. If I am talking about the relative growth, here you can see the rate of the growth with respect to the initial size. The growth of the rate with respect to the initial size is called as a relative growth. Okay. The relative growth we are telling in the percentage. Okay. If I compare the growth of the leaves for the initial growth, okay, initial growth I'm comparing, it means, for example, you can take a leaf A, okay, leaf A here you can see it has been increased 100%, 100% it is increased its length, yes or no? Before it is 5 centimeter and after that also it is 5 centimeter increased, means it is 100% increase, right? And if you take B leaf means it is 50 centimeters square before and it is increased only a 5 centimeter square. 5 centimeter square. Now it is 55 centimeter square means how much it is increased? Only at 10%. Right. Only 10% increased when compared with the initial size. When compared with the initial size, it is 10%. Always remember, if they ask about absolute growth means it is a growth not comparing with the initial size, whereas the relative growth means comparing, relative comparing. Okay. Fine. You all know the conditions for the growth. What are the conditions we require? We require a water, light, nutrients, oxygen, temperature, and the gravity. Right. These all the things are important for the growth. With these all the things only the plant is going to be grow. And let us talk about the some important topics, guys. Here I want to tell, just observe here what I am telling. Then we'll go to the topic. Okay. What do you mean by differentiation? See here, guys. Meristematic cell is there. Meristematic, remember, meristematic cell is there. Okay. This meristematic cell is given to the, that is going to be divided. It is going to be divided. Now, this divided cell converted into the parenchyma. Parenchyma. You all know that parenchyma has its own function, that is the photosynthesis and storage of the food, right? Means this meristematic cell giving to the parenchyma, that is called as differentiation. Differentiation. Remember, it is differentiated. Okay. So now what will happen? This parenchyma cell is there, right? Now this parenchyma cell, can you just go back to the anatomy of the flowering plant, guys? In the anatomy of the flowering plant, you can see that cambial ring is there. Cambial ring is going to be formed, right? So here you can see this is a pericycle. Inside that vascular tissue are there, right? Vascular tissue are there. And between that there is a parenchyma cell there, right? Parenchyma cells, 1 minute. 1 minute. Yeah. So between there is a parenchyma cells are there, right? Parenchyma cells. Here you can see parenchyma cells, and here you can see xylem is there, and here phloem is there. Between the xylem and phloem, there is a one cambium is there, right, guys? Which is that cambium? Can anyone tell me? Which is that cambium, guys? Between the xylem and phloem? Intrafascicular cambium is there between them, right? Between them, intrafascicular cambium is there. But you can see that formation of the cambial ring is going to be taken place, right? Formation of the cambial ring. On the time, what will happen? There are the parenchyma cells is there. These parenchyma cells is going to be convert into the interfascicular cambium, interfascicular cambium, this parenchyma. This then it is going to be form a cambial ring, right? Cambial ring is going to be formed. Yes or no? You know that cambium ring is undifferentiated. Cambial ring is undifferentiated mass of cell, right? This is a meristematic cambial ring now, right? Now this part is called as, you can see that the parenchyma is there now between here. The parenchyma cell is differentiated into the interfascicular cambium, right? Conversion of that parenchyma into not intra, interfascicular, fascicular cambium is called as, what do you mean by it is called as, guys? Can you tell me what happened here? Now the parenchyma loses its function or gain its function? The conversion of parenchyma into interfascicular cambium is called as what? I need answer from you guys. Whether it is differentiation or redifferentiation. It loses its function. The parenchyma loses its function, right? It is no way now it is acting as a photosynthesizing or storing the food. It is get de-differentiated. De-differentiation. It is called as de-differentiation. Okay. So now what is happening? Okay, I will tell you. This interfascicular cambium, you know that cambial activity is going to be formed inside that cambial ring, inside near the pith. Near the pith, it is going to be differentiated as secondary xylem, and outside the pith means outside periphery region, it is going to be act as the secondary phloem, right? You know that xylem and phloem is differentiated cell, right? Now what is happening now? Interfascicular cambium is converted into the xylem and phloem, right? What is happening now? Differentiation, de-differentiation we studied. And what is another process? What is happening now? The same thing. Differentiation already happened. Very good. It is undergoing again redifferentiation. Very good, Puneet. Very good, Raju. It is redifferentiated. Redifferentiated, right? This process is nothing but differentiation, de-differentiation, redifferentiation. Kindly you have to remember example. Very, very important. Okay. Let us study this. Okay. I think no need to explain this. You can go to this slides. Okay. You can see here the secondary de-differentiation example is secondary xylem and phloem. Okay. And remember here, de-differentiation example is interfascicular, not intra. If you are given intra, then you will lose the 4 marks. Okay, then coming to the development. So here we can see that in the development, mainly the plasticity. What do you mean by plasticity is, you know that the plant is growing in the different environment, right? In the different environmental condition, the plant leaves is going to be given different structures. Okay, that is going to be called as plasticity. Kindly remember this statement, it's important. Different kinds of structure in response to the environment or phase of the life, that is called as plasticity. Okay, mainly in the exam, if they ask about the, can you give me the heterophily in based on the phases, mainly phases. If they ask about the phases, heterophily, the examples are cotton, coriander, and the larkspur. Is the example for phases, based on the phases, juvenile phase, and in the adult phase, the coriander structure and the larkspur structure is going to be changed. Okay, you can see here is the larkspur. Okay, this is an example for the phase. Okay, then coming to the heterophily due to environment. Heterophily due to the environment, if they ask me, the example only for this is buttercup. In the second B, they are given the buttercup. You can see that in the water habitat, it will form the spiny-like structure in the water condition. Buttercup is example for the environment heterophily. Remember this. Okay. Then coming to the growth factor, which is controlling the plant. The growth factor is important. Mainly the intrinsic factors are genetic factors and the plant growth hormones. Then extrinsic factors we already studied: light, temperature, water, oxygen. These all are the important as the extrinsic factors. Now let us study about mainly plant growth regulators. Okay, the plant growth regulators I already told. There will be a natural. In the natural, the examples are the natural phytohormones we will call as. The synthetic, as we cannot extract the natural one, so that's why we are going to use the synthetic one. Okay, naphthalene, acetic acid, you can see this is in the case of auxin. Okay, we will study about this all the things. Oxygen. Coming to the plant growth regulator, there are two types. One is the plant growth promoters. Promoters, you all know that they will act positively, right? They will act positively. They increase the growth of the plant. Examples of the growth promoters are auxin, gibberellin, and cytokinin. Okay, you have to remember ACG. Okay, ACG is going to be act as a promoter. And coming to the inhibitor, ethylene and abscisic acid act as a inhibitor. You have to remember here, ethylene sometime act as an inhibitor and sometime it act as a growth promoter. Ethylene, okay. Ethylene sometime act as a growth promoter and sometime act as a promoter also and inhibitor also. Okay. Then if it is promoting means what are the function it is going to do? It is going to do the cell division, cell enlargement, pattern formation, anthropic growth, flowering, fruiting, and seed formation. And if it is inhibitor means what it has to inhibit. For example, just imagine, guys. Uh, there is a no environment condition is worst. Okay, there is a high temperature is there. Okay, then you have to remember there is no water availability is there. Okay, on that time, just remember the plant is going to be produce the flower. The plant is going to do the flowering, or it is going to increase its length. Just imagine it is going to be do the flowering and fruit is going to be formed. No, right? It is not going to be formed because that is the condition is not suitable for the growth. There is some hormone is working, that is arsenic acid. Mainly what the arsenic acid will do, it will close the stomata and it will not promote the growth of the plant. Okay, that is called as the stress hormone. Mainly we'll study about that during the wound and stress and dormancy condition, and during the abscission, the mainly hormone involved here is the growth inhibitor hormone, ethylene and the arsenic acid. The synthetic hormone you have to remember NAA, naphthalene acid, 2,4D and 2,4,5T. You have to remember this all the things. Let us talk about the first, that is the auxin. You all know that auxin discovery, the scientist is important, on which they have worked is very, very important. Okay. The first growth hormone discovered is the auxin. Okay. The first who worked on this is the Charles Darwin and his son. They worked on the canary grass. Okay. They worked on the canary grass and they told that phototropism is happening because of the coleoptile, which is present on the stem, where it is going to be bend the stem, bend the shoot. That is mainly because of the phototropism. There, the hormone is present, that is nothing but the auxin. Auxin is helping in the phototropism. Okay, remember that. And it is a site for the responsible for the bending. Okay, remember. And the finally, first auxin was discovered by the W.F. Wendt. Okay, Wendt on the coleoptile tip of the oat seedling. Remember this. W.F. Wendt and coleoptile tip of the oat seedling. This type of the question you will get in the match the following. Fine. And you know that auxin is also isolated from the human urine, and IAA, indole acetic acid and indole butyric acid have been isolated from the plant, and the naphthalene acetic acid and 2,4D means 2,4 dichlorophenol acetic acid are the synthetic form. In the exam, they may ask you, they will give the auxin name. Okay, they will give the auxin name, and in that they will ask you which one is the synthetic one. In the synthetic one, you have to remember is the naphthalene acetic acid and 2,4D is the synthetic one we are using in the agriculture. Okay. Then the functions are very important, guys. These will initiate the rooting. You have to remember the auxin and cytokinin are used in the culture. Mainly, the auxins are used to increase the root. Okay. Whereas the cytokinin is helping in the shoot formation. Auxin is helping in the rooting. Remember, some will get the confusion over here. And auxin promote the flowering in pineapple. Don't confuse with the synchronizing the flower set in pineapple. That is a different function. Okay. The promote the only flowering in pineapple means that is auxin. Okay. Then it will prevent the fruit and leaf drop at early stages. At early stages, the fruit is not going to be drop, right? And the flower is not going to be drop, right? That is because of auxin. Okay. And whenever the fruit and whenever the flower has been ripened, whenever the fruit is ripened, then only it is going to be fall, right? That is mainly because of the ethylene. Remember, it is going to be prevent the leaf and fruit drop during the early stages, and it promotes the absorption of the older and mature leaves and the fruit. Ethylene also working, and also auxin is also working here. Promote the absorption of older mature leaves and also the fruit. Okay. And the apical dominance, you can see, guys, the plant is goes on increasing its length, right? There is no lateral branching, you can see. Means there is a auxin is working on the shoot, so that's why it is inhibiting the lateral growth. Remember, okay? The apical dominance is mainly due to because of auxin. Remember, okay? And auxin also induce the parthenocarpy. You know that what is parthenocarpy? Seedless fruit. Seedless. Seedless fruit. For the seedless fruit, we are going to be use the that is synthetic auxin. Okay. And also 2,4-D is used as a herbicide. We will use this 2,4-D as a herbicide. Okay. And it is not going to be eliminate the grass. Remember, guys, it is going to be eliminate the monocot, everything, other thing, but it is not going to be eliminate the grass. Okay. And the xylem differentiation and also helps in the cell division. Okay. And you all know that what is the apical dominance, no need to explain here. Then coming to the gibberellin. Mainly the gibberellin, you can see that Bakane disease. While studying the Bakane disease, we identify the gibberellin. Mainly the Bakane disease is due to the fungus, that is Gibberella fujikori. Remember, Gibberella fujikori. Kurosawa is worked on that. He treated the uninfected rice seedling with the sterile filtrate of this fungus. Then he saw that there is an increasing in the length of the plant and appearance of these symptoms. Okay. You have to remember mainly the gibberellinic acid. Okay, what are the function here mainly is the gibberellin is going to be increase the length. It is going to be increase the length of the axis. Here you can see a grapes. Okay, increasing in the length of the axis. Here the grapes is increased its length due to the application of the gibberellin. Remember, in the grapes and in the apple, mainly due to the gibberellin. Okay, and the fruit is going to be elongate. That is apple and the grapes. Okay, and delay the senescence. Delay the senescence. It will delay the aging process. Okay, and in the malting process also we will use this gibberellin. And length of the stem is going to be increased in the sugarcane. It is very important. Okay, and it will increase it. This is also important, guys. In some of the MCQs you will get that. Spraying the juvenile conifers, spraying this gibberellin to the juvenile conifers, it will hasten the maturity. The maturity is going to increase rapidly in the juvenile conifers. Okay, remember this. This is an important point. And mainly the bolting. What do you mean by bolting? Means before the flowering, it has to be changing, right? Before the flowering, the internodal elongation is going to be happen, right? That is called as a bolting. Mainly the bolting hormone is gibberellin. Gibberellin, remember. Mainly in the rosette, that is in the beetroot and in the cabbage and in the rosette habitat, we will use this gibberellin for promoting the bolting. And coming to the cytokinin discovery is by the Skoog and his co-workers. Mainly you have to remember here, Skoog and the Miller identified the substance that is kinetin. Kinetin is responsible. Okay, kinetin is responsible for the cell division and proliferation. Kinetin is also nothing but the cytokinin. Remember, and cytokinin has a specific effect on the cytokinesis. You all know that. What do you mean by cytokinesis? Division, cell division. Cytokinin, cytoplasma division is nothing but the cytokinesis. Cytokinesis involved in the cell division. Cell division, mainly in the shoot formation. Shoot. Okay, for example, the calyx is there, right? For the calyx, if we give the cytokinin means it is going to be form the shoot. Okay. One minute. Okay, guys, fine. Then coming to here, kinetin from the autoclaved herring sperm. Here you will get the MCQ light. Which one is identified from the autoclaved herring sperm? Okay, herring sperm. Here you will get the confusion between kinetin and zeatin. You have to remember kinetin. Or in the exam, they may ask about the cytokinin. Which hormone is identified from the autoclaved herring sperm? They will get the kinetin in the mind, but you will not get the cytokinin. Just remember cytokinin, that is nothing but the kinetin. Okay. And from the corn kernel and from the coconut milk, the identified is zeatin, not the kinetin. Remember that. Okay. And the natural kinetin also synthesized in the region where a rapid cell division is going to be happen. You know, wherever the rapid cell division is going to happen, that is in the root and in the shoot and in any some birds, you can see there is a cytokinin is present there. Okay. So this is about the cytokinin. Mainly the function of cytokinin is helps in the production of new leaves, chloroplast, and in the fruit formation. And this is very important in the cytokinesis, lateral shoot formation. You know that apical dominance is done by the auxin, right? Wherever the apical dominance is happened, right? For that shoot, if you apply this cytokinin, means there is a lateral bud is going to be happen. For example, you can see there is a apical dominance is happening, right? What you can do is either you can remove this shoot part, okay? Either you can remove the shoot part, then a lateral bud is going to be happen, or another thing you can do is in this shoot part where is this auxin will be there, right? You can apply cytokinin. If you apply cytokinin, means there the lateral bud is going to be formed. Okay, just remember this. The same thing we will do in the tulsi plant also, right? The elders in the home, what they will do, they will pluck the shoot of the tulsi plant, because they will tell that the branches will come nicely. But if they don't know what is there myth there, right? There is a thing is, by removing the shoot part, there is the auxin is there where it is inhibiting the lateral growth by the dominant apical dominance. By removing that shoot part, by plucking that, there is a lateral growth is happening, because there will be a no auxin will be there. That's why. Okay. Then there will be adventitious shoot formation. Adventitious shoot formation is done by the cytokinin. I have told you. The callus, if you kept the callus in the culture medium, this is a culture medium. Okay, culture medium. For the culture medium, if you give the cytokinin means there is a shoot formation is going to be happen. Okay. Then cytokinin is also helps in the apical dominance. Now only we studied, and it will delay the leaf senescence. Okay. And ethylene, you know that it's a gaseous substance. It hastens the ripening in the oranges and ripening of the fruit. Okay. And here you can see the mainly the triple response. Triple response is by the mainly the ethylene. What are the triple response? Horizontal growth of the seedling, swelling of the axis, and apical hook formation in the dicotyledon seed, mainly due to the ethylene, and it will promote the senescence and the abscission. Ethylene is highly effective in fruit ripening, because we will using the ethaphone here. Ethaphone. Okay. And it will break the bud dormancy and initiate the germination. Ethylene promote the rapid internode petiole development in the deep water rice plant. Remember, guys, if they talk about the elongation or increase in the length in the sugar cane, means that is gibberellin. If they ask about that internode elongation in the underwater rice plant, means example is ethylene. Underwater rice plant, don't get confusion. And also it is helping in the synchronizing, not flowering, synchronizing and flowering in the pineapple. Okay, in synchronizing and flowering. If they ask about only flowering, auxin. Synchronizing in the fruit set means that is ethylene. And also the root growth and the root hair formation, and flowering in mango, very important. Flowering in mango. And you know that ethaphone is increasing the fruit ripening. Then coming to the abscinic acid, it is also called as inhibitor B, abscissin 2, and the dormine. Okay, you have to remember this all the three names. And it is antagonistic to gibberellin. they will ask in the examination which hormone is acting as an antagonist to gibberellin. The example is arsenic acid. The mainly function of the arsenic acid is plant growth inhibitor and inhibitor of the plant metabolism, abscission and the dormancy, inhibit the seed germination, closure of stomata, and seed to withstand the desiccation and other unfavorable condition, and also antagonistic to the gibberellinic acid. Okay, these all are the main function of the arsenic acid. Let us answer to this question, guys, if you understood me. Okay, here is the first question. That is the match the following question. Arsenic acid, ethylene, and gibberellin. Here you have to do the find the function of that. I'll launch the poll. You can answer me in the You can answer me in the chat box also, guys. I have relaunched the poll. You can answer me in the poll also. Yeah, very good, very good. I want answer from the others also. Answer me, guys. Answer me fast. Which one is the correct? You can answer me in the chat box also. I'm sharing the result. Yeah, 100% is given by only the one student have answered for this question. Okay, 1 minute. What about the others, guys? Yeah, the correct answer for this question is option C. Okay, how it is C? Kindly see here, guys. Arsenic acid. Well, let us start from the arsenic acid. We know that arsenic acid is helps in the seed to withstand the desiccation, dormancy, right? For the A, it is 2 means. And ethylene, what is the function of ethylene here? Promote the flowering in cucumber. Then A should be 2 and B is 1. A is 2 and B is 1 only in this. And coming to the C, gibberellin. What is the function of gibberellin? Nutrient metabolism, 3. Right. And what is the function of cytokinin? Cytokinin is helping in the sorry. Gibberellin is a 4, right? Gibberellin is a 4, that is bolting hormone, beet and cabbage. Then cytokinin, you can see here, cytokinin is the cytokinin is 3, helps in the nutrient mobilization. Right? The correct answer is C, not D. C. Okay. I got the answers. Yeah, very good, Sai Charan. Kindly go to that slide. Okay. Cook observed that callus proliferated from the internodal segment of the tobacco stem when auxin was supplied with one of the following, except. Okay, so here you have to see that Skoog has taken the, sorry, I have given the correct answer, guys. Sorry. C is the correct answer. See here, the Skoog has taken the callus, okay, he's taken the callus, and in the nutrient medium, auxin is there, okay. Along with the auxin, he is taken the extract of the vascular tissue and extract of the yeast and coconut milk. In this, all the thing, cytokinin is there. Cytokinin is there. Okay. Only the abscisic acid is not there. Remember. Okay. Given below are the some statements about the plant growth regulators. Okay. Kindly you can answer to this. Meanwhile, I will launch the poll, teacher NPS poll. You can give me your feedback. Okay. Yeah. I'm launching the teacher feedback, guys. You can give me your feedback. Meanwhile, you can, after that, after giving the feedback, you can see the question and you can answer to this question. Okay. So the question is, given below are some statements about the plant growth regulators. Okay. And which one of the statement is correct? Okay. You have to give the correct statement here. You can answer in the chat box also. Okay. Let us answer. All the gibberellinic acids are acidic in nature. You have to choose the correct one. Okay. And auxins are antagonist to the gibberellin. And zeatin was isolated from the coconut milk. Ethylene induced the flowering in mango. Arsenic acid induced the parthenocarpy. So the correct answer is A. That is ACD. They are asking about the correct statement. All gibberellinic acids are acidic in nature. Yes. Then coming to the auxin is antagonist to gibberellin. No, auxin is not antagonist to the gibberellin. Cytokinin, cytokinin, right guys? Cytokinin is antagonist to the auxin, whereas the gibberellinic acid is antagonist to what? And zeatin was isolated from the coconut milk. That is correct. Then ethylene induced the flowering in mango. That is correct. Then arsenic acid induced the parthenocarpy. No. Auxin is going to be induced the parthenocarpy. Yes or no? Yes. Very good, Sai Charan. Very good, Nili. Yes. Correct. Correct option is A. Let us go to the next question. You can answer to this question. Auxin is used by gardeners to prepare a weed-free lawn. Auxin. Okay. You know that 2, 4D. 2, 4D is used by the lawn. Okay. But no damage is caused to the grass. No damage is caused to the grass. Why? Why? I'm getting the answer. Very good. Very good, Nili. Yes. What about the others? Yeah, you have to remember here, guys. It is not going to affect the very good, Nili Shruti. Very good, Sai Charan. Very good, Shreya. Yes. It does not affect the mature monocotyledon plant. That's why the correct answer is C. And here you can see formation of interfascicular cambium. Remember, guys, interfascicular cambium from fully developed parenchyma cell is an example for. Now only I explained in clearly. I will see whether how many of you will answer correctly. If you know the answer, you can give me the answer in the poll. I'm relaunching the question, guys. Poll, kindly answer me. Very good, Neeli. What is happening? Formation of the interfascicular cambium. Just remember, guys, the meristematic tissue is there. Meristematic tissue is there, right? Meristematic tissue will give the parenchyma cell by the differentiation, right? Then parenchyma cell is undergoing the interfascicular cambium by the differentiation. Then again, this interfascicular cambium is going to form the secondary xylem and secondary phloem by the process redifferentiation. The correct answer for this is option C, that is de-differentiation. Right? One more question for you guys. Spraying the sugarcane crop with which of the following plant growth regulators increase the length of the stem, thus increasing the yield? Which hormone is used for the sugarcane? Very easy question. Sugarcane. Sugarcane. Think once again, guys. Very good, Nili. Very good, Sai Charan. Very good, Shashank. Very good, Jamlil. Very good, Shreya. See here, guys. Mainly concentrate on a plant hormone. Other topics are very easy, right? Kindly concentrate on some characteristic and some functions will be confusing. So that's why you have to concentrate on the more hormones. Okay. Option D is the correct answer. And here is the match of following. You can answer to this question and you can leave the class. Okay. If you are having the time, kindly be there. Let us solve some another 1 or 2 question. So these are based on the functions, guys. You can match the following: auxin, gibberellin, cytokinin, and the ethylene. Which one is the correct answer? Yeah, I'm ending the poll. I'm relaunching the question. 1 minute. Yeah. Yes, Nili. Yes, Shreya. So many will get the confusion between the synchronizing the flower set in pineapple and the pineapple flower. Kindly remember, guys, flowering is the auxin function, flowering in the pineapple, and synchronizing the flower set is the example is in the ethylene. Okay, and the flowering in the mango is the ethylene. Remember, okay. The correct answer for this is B. Very good, guys. Option B is the correct answer. So here, A is the 4. A is the 4. Promote the flowering in pineapple. Very good. A is the 4. Then gibberellin. For the gibberellin, it is increasing the length of the grape stalk. Yes, it is the 2. 2, okay, 2. Then coming to the cytokinin, it is 2. That is overcoming the apical dominance. And the ethylene is the one that is promoting the female flower formation in the cucumber. Ethylene. Ethylene is in the cucumber in the flowering in the cucumber and in the mango also. Spraying of which one of the following phytohormone in the juvenile conifer helps in the ascending the maturity period that leads the early seed formation. Early seed formation. I am relaunching the poll. Answer me. And in the chat box also you can answer me, guys. Very good, Sai Charan. Very good. What about others? That's why, guys, I have underlined it there. You have to remember this point. If you don't know, kindly make this point because it is more confusing, right? Kindly make this point. No, it is the correct answer is cytokinin. In the cytokinin we studied, guys, this question. The correct answer is B. Kindly see here. I will go back to the question. Okay, 1 minute. This is the questions. We will go to the cytokinin. Okay. There I have underlined it and I have told you also. 1 minute. Yeah, see here in the cytokinin. Okay, spraying the juvenile cohorts with the gibberellin. 1 minute, guys. In the gibberellin, the correct answer for this is gibberellin. 1 minute. But why it's zeatin? Sorry, guys. The spraying the phytohormone. 1 minute. Spraying of the phytohormone juvenile conifers with ascending the maturity period. Here, juvenile, right? Juvenile conifers with the ascending the maturity period. The correct answer is gibberellinic acid. Gibberellinic acid, not zeatin. Okay, not zeatin. That is the gibberellinic acid. So like this, you will get the confusion. So kindly make this point, make a note of this point and check with the gibberellinic acid. Okay. Gibberellinic acid. A. Spraying of the juvenile conifers with the maturity period is going to be Aston. Here is the gibberellinic acid. Have you understood this, guys? Can you make a note of this? Okay, fine. This is about the today's class, guys. The mainly the phytohormone functions will be confusing. While coming to the next class, I will bring some mnemonics or I will add the mnemonics in this PPT only. You can go through that. Okay, fine, guys. Let us end the class. Where yes, Shreya, I will show. So here we got confusion, right? Sorry, guys. Gibberellin is the answer. Okay, not zeatin. Oxen slide. 1 minute, Shreya. Here in the gibberellin we studied, right? Function of the 1 minute. Yeah, gibberellin. In the gibberellin, we studied that, guys. Juvenile conifers is going to be after the maturity period of. I told you it's important also. Okay. This is the gibberellin. Okay. So this is the oxen slide. So yeah, you asked for oxen slide, right? See here. Shreya, have you seen right this slide? You want another slide? Yes. Fine, no? Share is the first slide of the oxen, and this is the second slide. You can go through the notes also. Okay. Fine. Can I end the class? Shreya, do you have any question? But can I end the class? Yeah. Thank you so much. We'll meet in the next class. Okay. Thank you, guys.